You guys ready to move on to the next level? So the ATP that's produced in glycolysis is called substrate level phosphorylation. Substrate level phosphorylation happens in the cytosol. So if we are in an aerobic cell and there's plenty of O2 around, so we will then take our pyruvate and shuttle it off to the CAC cycle, the citric acid cycle, with the hope of making a boatload more ATP, right? So we take our pyruvate. It's got 3 carbons. So if you look, what's my entry point into the citric acid cycle? Acetyl-CoA. And so here we have this little tiny step, and I'm being facetious, of turning pyruvate into acetyl-CoA and carbon dioxide. So then we're going to form some carbon dioxide. Cool, this is actually a giant conglomerate of enzymes that actually carries this out. So pyruvate dehydrogenase. Where have we seen the word dehydrogenase before? Yeah, it was the step in glycolysis that was an oxidation. So in fact, pyruvate going to acetyl-CoA and CO2 is indeed an oxidation. And if there's an oxidation, what else has to happen? There's a reduction. What's my big cofactor getting reduced here? Yeah. So most of the time it's NAD reduced to NADH. We'll see one case in the citric acid cycle where it's FAD instead of NAD. So, but in this case, it's going to be that.
So is this one the 3 NADHs that are counted?
So this is, so where are my NADHs here? So one there, one there, one there. Those are your 3 from citric acid cycle. This is separate.
We're not even.
Good. Yeah, so we're not quite in the citric acid cycle yet. So one thing to note, this is going to be one of the places where I save some time. So there are some players here. Thiamine pyrophosphate. So heard of thiamine before? What's thiamine? One of B vitamins. So he's involved in the process here. So we just saw that NAD getting reduced to NADH is involved in here. It turns out that this is not a one quick little step thing. It's like 4 or 5 steps. Lipoic acid is involved. So and your flavonoid cofactor is also involved, FAD. Lots of players involved here. So and also some regulation that I'm going to leave alone. So this is one thing you want to definitely go back on. So it's rather complex for just drawing one little quick arrow. So your pyruvate gets bound to TPP, so with a lovely thioester, which gets handed off to lipoic acid. So coenzyme A is also involved in the process. So we'll get a thioester forming along the way. Lipoic acid gets oxidized and reduced in handing off. So our pyruvate off to the next one down the chain, forming the acetyl-CoA when it hands it off to the coenzyme A. So lots of stuff going on here, lots of regulation going on here. I'm leaving that on you. And we're going to jump into the citric acid cycle. So we have 40 minutes, 42 minutes. I'll use all 42. Cool. It's your acetyl-CoA. This is your feed point into the citric acid cycle. Acetyl-CoA has 2 carbons. So what am I going to combine my acetyl-CoA with? Oxaloacetate, which we saw a little bit earlier as part of the gluconeogenesis. So and how many carbons does oxaloacetate have? 4. And so by the time you combine acetyl-CoA with oxaloacetate, you end up with 6 carbons in your citrate. So here's the deal. You need to know your entry points. Acetyl-CoA reacting with oxaloacetate. So I would memorize this thing if I were you in stages. Work on the intermediates first. So start there. then work on where carbon dioxide, NADH, so FADH2, and your 1-GTP, which really gets converted into an ATP, work on where those are occurring. So, and then maybe after that, go through the enzymes. And if you notice the enzymes with a red asterisk, those are your major regulation points for the citric acid cycle. So, but if you try and memorize this all just one time, go around and around and memorize more and more, you're going to burn yourself out. Memorize it in stages. So if you look, so citrate, isocitrate, and alpha, alpha being the key here. So the CIA is after you, so you know. So bad news is so is the Secret Service. They're after you as well. So if the CIA and the Secret Service are both after you, this should make you fuming mad. Sweet. So start there. I don't have lovely little, phrases for everything, but I do have one for this. So the CIA and the Secret Service are after you. That should make you fuming mad. Cool. Once you've gone there, so I would keep track of where your carbons are. Every place you lose a carbon, notice we lose one from isocitrate to alpha-ketoglutarate. Where's that carbon go? So that's your CO2 being given off. So and if your CO2 is given off, usually that implies that you had a carboxylate that was decarboxylated. In a carboxylate, how many bonds does carbon have to oxygen in the carboxyl group? How many bonds to oxygen from that carbon? 3. 1, 2, 3. So if we're undergoing decarboxylation, how many bonds to carbon from oxygen now? 4. Very often implies an oxidation. And if it implies an oxidation, what must happen at the same time? Reduction. Sweet. Notice where I produced CO2, guess what got reduced? NAD. Sweet. The next time we produced CO2 is when we went from alpha-ketoglutarate, which is 5 carbons, to succinyl-CoA, which is only 4 carbons. Lost another CO2. That, again, was oxidation. And so we need NAD to get reduced along with it yet again. Cool. Now we're down to 4 carbons. Are we going to lose another carbon anywhere? Not through this cycle. So all the way to oxaloacetate, we stick around with 4 carbons. So a couple things to know. So in this step, we end up with succinyl-CoA. So big thing you should know about succinyl-CoA. What kind of linkage? That's a high energy linkage are we talking about here? It's a thioester. Thio means what? Thio, sulfur. And so notice so some sort of thioester here. So it's going to be analogous to your normal ester, but with a sulfur instead. So when succinate's bonded to CoA here, if you will, succinyl-CoA, it's got a very high energy thioester bond. So, which is good. What usually happens when you have a high energy bond? You're about to get some energy out of it. And what kind of energy do we get? We get some GTP out of it. Sweet. So, and GTP is functionally equivalent to ATP. And it's not just that it's functionally equivalent, it's that there's an enzyme that converts GTP to GDP and transfers that phosphate to ADP, which becomes ATP. And so it's not just functionally equivalent. We saw that GTP was used by a kinase earlier instead of ATP, but it also essentially turns into ATP end result. Cool. Succinate to fumarate. What is that? FAD turning into FADH2. What's happening to my cofactor here? It's being reduced. Succinate to fumarate must be an oxidation. If you notice, this is the one oxidation around the chain. So the first one we've seen anyways, that did not involve giving off CO2. So it's not that when you produce CO2, those are the only oxidations. So those definitely are oxidations and evidence that it happened, but they're not the only ones. We see our next oxidation step right here to fumarate. So fumarate to malate, it's just a hydration. So adding water to the molecule, essentially, H and OH. So, and then finally, malate to oxaloacetate must again also be, so one more redox reaction. malate to oxaloacetate, one last oxidation in the cycle, which is why NAD again gets reduced. So if you notice, your food just keeps getting oxidized and oxidized and oxidized. Essentially this in case you're glucose. And we just keep oxidizing it and oxidizing it as often as we can. In this case, in a cycle, why do we do that again? Let's not lose the big picture. So, and where does that energy go when we oxidize our glucose? Where is it temporarily stored, so to speak? In our FADH2 and our NADH at this point in time. So, and where can I get the energy out of those? Electron transport chain. So eventually, in the next section of material for the next exam, we're going to shuttle off all this NADH and FADH2 we're making, and we're going to ship it off to the electron transport chain and get a boatload of ATP out of it. All right, so now have all the intermediates in place. We've now talked about where we get the NADH, the FADH2, where carbon dioxide is evolved, and the one step that actually gets us an ATP. So one thing to keep in note, what did we shuttle into this citric acid cycle? Acetyl-CoA, where did that come from? Pyruvate, where did pyruvate come from? Glucose. So keep in mind, how many pyruvates do you get for one glucose? 2. So everything we do here, if you're looking at it per pyruvate molecule, that's what it's set up for. If you're looking at it per glucose, double everything. So be careful. They can ask the question either way, per pyruvate or per glucose. And again, glucose yields 2 pyruvates. You got to double everything. So if you notice, how many NADHs do we get in the whole cycle? 3. And how many FADH2s? 1. But if you looked at per glucose, for the citric acid cycle, how many NADHs would you get then? 6. And how many FADH2s? 2. Notice we only get 1 GTP or ATP. You could look at it either way. So, but if you do it per glucose, it would be 2, not just 1. So again, be careful. Really look at that question. Is it per pyruvate, which is 1 time through the cycle, or is it per glucose, which is 2 times through the cycle?
So if you were to ask what the net NADH or FADH2 production is from the complete combustion of one molecule of glucose, what would that be?
So let's look. If you go back to glycolysis, so how many oxidation steps did we have in glycolysis? In fact, we'll go back there real quick. Where's your big oxidation step in glycolysis? Good, right there. That's it though, right? That's your only oxidation step. Notice the key you got to remember is though, did it happen before or after the split of our 6 carbon? After, which means it happens twice. So there's 2 NADHs. Okay. So did we get any other NADHs before we ever got to citric acid cycle?
Yeah.
Where at?
Pyruvate to acetyl-CoA.
So pyruvate to acetyl-CoA. How many NADHs do we get there?
2.
Awesome. For 1 glucose, which gave 2 pyruvates, you'd get 2 in converting 2 pyruvates to 2 acetyl-CoAs. So how's our tally, Justin? How many NADHs we got so far? 4. And if both pyruvates go through the entire cycle, so after turning into acetyl-CoA anyways, both acetyl-CoAs go through the entire cycle, how many more NADHs do we get? 6 more, for a grand total of 10. And how many FADH2s? 2. Sweet. How many ATPs did we get in glycolysis from 1 glucose? Net. 2. How many more do we get from the citric acid cycle? Cool. We get a couple more. Again, 1 for each cycle, so 1 for each acetyl-CoA that goes through. Good times. So if you notice, it's one of the things that makes this stuff a little bit more difficult too, is you got to see the big picture, and then you got to zoom in and look at the little details of each step at the same time. So, and a lot of times, we're so focused on the details, we forget the big picture. So, awesome. So let's look at your big focal points for citric acid cycle control. So we're not going to go through all of the enzymes. So although I don't think that would be necessarily the worst thing in the world to know, but you definitely need to know the ones under regulatory control. So citrate synthase. So what did citrate synthase, what do you suppose he does? Yes, he makes citrate. And what does he make citrate from? That's the first entry step, oxaloacetate and acetyl-CoA. And so in this case, that's a big point of control, starting the cycle, it turns out. So, and it's activated by ADP. Does this make sense? Yeah, evidence that your cell is under kind of low energy conditions, if you will. So it's also activated by acetyl-CoA. Does that make sense? Yes, because that's the, or at least one of the, reactants in that step. So again, we're turning acetyl-CoA and oxaloacetate, combining them into citrate. And again, if I got a big pool of acetyl-CoA, might be a good time to get this citric acid cycle going, make some energy. So it's inhibited by citrate. Does that make sense? Because that's the product. That's product inhibition. So in fact, succinyl-CoA, it isn't the product of that reaction, but it's a little further down the chain, right? So it doesn't, it's not, wow, didn't see that coming kind of a thing. So it makes sense. I might not have predicted it, but it does make sense. It's further down the chain. It's not the immediate product, but it's kind of like product inhibition. So, and then to a much lesser extent, ATP and NADH are inhibitors as well. I don't know how important this really is to know. Does this make sense?
ATP does, but the NADH I don't understand.
Ooh, NADH is as good as gold. Because what am I going to do with that NADH? I'm going to ship it off the electron transport chain and trade it in for a boatload of ATP. So it also is a high energy compound and evidence, if you got a lot of that you got a high energy situation going. So it's not energy yet, or at least it's not ATP yet, but it will be. Isocitrate, dehydrogenase. When you hear the word dehydrogenase, what should you think? Oxidation. So in your isocitrate dehydrogenase, isocitrate to alpha ketoglutarate. And again, we're losing a carbon to CO2 in this step. So in this case, it's activated by ADP. Does that make sense? Yeah, in fact, if you notice, all three big regulation points in the citric acid cycle are all activated by ADP. So that makes perfect sense. So in this case, it's also activated by calcium. Memorize it. So inhibited again in a minor way by ATP and NADH. Again, both evidence that you got a high energy state in the cell. So again, it makes sense. We don't need to do a lot of citric acid cycle if I already have lots of energy, or at least high energy compounds in the cell. So alpha ketoglutarate dehydrogenase. What do you suppose dehydrogenase again? Whoop, wrong way. Oxidation again. So in this case, your alpha ketoglutarate dehydrogenase, alpha ketoglutarate to succinyl-CoA, and again, losing carbon dioxide yet again. And if we look at our regulatory points, so again, we already said ADP activates, and again, I'm not actually even sure of the whole calcium connection here, so I'm sure it makes sense to somebody a little more skilled than me. So succinyl-CoA inhibits it. Does that make sense? Oh, go ahead.
Calcium from muscle contractions, is that showing that you're using up energy or something?
I don't know. I don't know if it's necessarily from muscle contractions, because notice those are the calcium channels and stuff like that. Maybe. So, but so honestly, I don't know. So succinyl-CoA inhibiting it. Does that make sense?
Yes.
Why does that make sense? Product inhibition. NADH and ATP, again, minor. So ATP and minor anyways, but again, high energy situations. Cool. So what's nice is these have a lot in common as far as their regulation. You got to remember which enzymes are regulated. So I'd know everything about this. Who's the reactant? Who's the product? What their structures maybe even look like. So, and then also all the regulation. So pyruvate carboxylase. Pyruvate carboxylase. We saw pyruvate carboxylase. We didn't see it as part of the CAC. What is that? Gluconeogenesis. It was turning pyruvate into oxaloacetate, which potentially could fuel the oxaloacetate that's here. So it turns out this cycle has a lot of entry points. There are a lot of things that I can turn into these different intermediates, which is good. So because this thing is what we call amphibolic. What's an amphibian? Land and water, kind of dual natures. So amphibolic is something that could be both catabolic and anabolic. Catabolism is when you're breaking your food, in this case, so your food down, your fuel. So whereas anabolic, like anabolic steroids, is where you're biosynthesis, we call it. So you're building stuff. Cell needs stuff, let's build it. So in this case. So it turns out that the way we've been talking about this is all catabolism. We're trying to get energy from our food. So, but it turns out that a lot of the intermediates here can be used to make things such as amino acids, things of this sort. And so if all of a sudden I'm doing this cycle and I shuttle off the alpha-ketoglutarate to make some amino acids, I just stop the cycle. So there has to be a way to replace it from something else. And so there are entry points and exit points. The same amino acid I might use alpha-ketoglutarate to make under some conditions for biosynthesis. Well, if I reach a high energy state, so that might be a good idea. I don't really need to do the citric acid cycle if I have lots of energy. But if I'm in a low energy state, I might not think I need those amino acids too badly anymore. And I might shuttle them back in to replace these intermediates and get the citric acid cycle going. So it's amphibolic. That's probably a word worth knowing.
Intermediates. What is that?
What is that word?
I cannot remember it.
So it's on your sheet. So anaplerotic. So when we replace these intermediates of any pathway, it doesn't just have to be this pathway, but of any pathway, when we replace those intermediates, shuttle them in from different places, anaplerotic. So it's on your handout there. So if you notice, I put them on there, like pyruvate to oxaloacetate by pyruvate carboxylase. That's an anaplerotic reaction. I would make a big note of that. So also transamination of aspartic acid and glutamic acid. So you can feed in, those are amino acids, by the way. You can feed in making some alpha-ketoglutarate, so, or your oxaloacetate as well. Couple other big things on here. Citrate. You can use citrate to make lipids. You can use oxaloacetate, as we saw in gluconeogenesis, to make glucose. So you can use oxaloacetate to make amino acids. So you can use alpha-ketoglutarate to make amino acids. So you can also use some of the intermediates here to make heme, so for red blood cells, as well as purines and pyrimidines for DNA and RNA. So lots of things the intermediates in here can also be used for than just simply making energy. So when the cell has lots of energy, we'll often start shuttling these off for biosynthesis.